The human immunodeficiency virus, or HIV, has cast a long shadow over the world. Since its emergence, it has claimed millions of lives, leaving behind a trail of grief and uncertainty. Yet, even in the face of this devastating disease, humanity has shown remarkable resilience. We have poured our hearts and minds into understanding this complex virus, developing treatments that have transformed HIV from a death sentence into a manageable condition for many. But the fight is far from over. The impact of HIV extends far beyond the individuals it infects. Families are torn apart, communities are devastated, and the social and economic fabric of nations is strained. The stigma surrounding HIV, fueled by fear and misinformation, continues to cast a dark shadow, preventing people from seeking testing and treatment. Yet amidst these challenges, there is hope. Scientists around the world are working tirelessly to develop a cure for HIV, and recent breakthroughs in gene editing, immunotherapy, and other fields have brought us closer than ever to this goal. HIV is a type of virus known as a retrovirus. Retroviruses are unique because they have the ability to integrate their genetic material into the host cell's DNA. This integration allows them to persist in the host for long periods, making them particularly challenging to eradicate. HIV specifically targets the CD4+ T cells, also known as helper T cells. These cells are a vital component of the immune system, acting as the orchestrators of the body's defense mechanisms. They help activate other immune cells, such as B cells, to produce antibodies and macrophages to destroy ingested microbes. Think of CD4+ T cells as the generals of our immune system. They coordinate the body's defenses against a wide array of invaders, including viruses, bacteria, and fungi. Without these generals, the immune system's response becomes disorganized and ineffective. But HIV is a cunning adversary. It has evolved sophisticated mechanisms to evade the immune system. One of its primary tactics is to hide within the very cells that are supposed to protect us. The virus inserts its genetic material into the DNA of these immune cells. This process, known as reverse transcription, allows HIV to hijack the cell's machinery, turning it into a factory for producing more virus particles. Each infected cell can produce thousands of new viruses, which then go on to infect other cells. This insidious process gradually weakens the immune system. Over time, the number of CD4+ T cells declines, leaving the body increasingly vulnerable to a range of opportunistic infections and cancers. These are infections and diseases that a healthy immune system would normally be able to fight off with ease. As the virus replicates and the immune system deteriorates, a person living with HIV may experience a range of symptoms. Early symptoms can include fever, fatigue, and swollen lymph nodes. As the disease progresses, more severe symptoms such as unexplained weight loss, chronic diarrhea, and persistent infections may occur. Without treatment, HIV infection progresses to AIDS, which stands for Acquired Immunodeficiency Syndrome. AIDS is the late stage of the disease, characterized by a severely weakened immune system. At this stage, the body is unable to fend off life-threatening infections and cancers, leading to severe health complications and ultimately death if left untreated. Fortunately, modern antiretroviral therapy, RT, has transformed HIV from a fatal diagnosis into a manageable chronic condition. ART works by suppressing the virus's ability to replicate, thereby preserving the immune system and preventing the progression to AIDS. With consistent treatment, individuals living with HIV can lead long, healthy lives. It's important to remember that while there is no cure for HIV, ongoing research and advancements in medical science continue to improve the quality of life for those affected. Education, prevention, and early treatment are key to managing and combating this virus. Understanding HIV and its tactics is crucial in the fight against this virus. By staying informed and taking preventive measures, we can protect ourselves and support those living with HIV. The development of antiretroviral therapy, or ART, marked a significant turning point in the fight against HIV. Before ART, an HIV diagnosis was often considered a death sentence with limited treatment options available. The advent of ART has transformed HIV from a fatal disease into a manageable chronic condition. ART consists of a combination of drugs that work in different ways to suppress the virus's ability to replicate. These drugs target various stages of the HIV life cycle, preventing the virus from multiplying and spreading throughout the body. This multifaceted approach is crucial because HIV can quickly develop resistance to a single drug. These drugs have been remarkably effective in reducing the amount of virus in the blood, known as the viral load, to undetectable levels in many people living with HIV. An undetectable viral load means that the virus is present in such low quantities that it cannot be detected by standard laboratory tests. This means that the virus is effectively suppressed, allowing the immune system to recover, and significantly reducing the risk of transmitting the virus to others. People with an undetectable viral load have a dramatically lower chance of developing AIDS-related illnesses and can lead healthier, longer lives. While RT has been a game changer, it is not a cure. The virus can still persist in the body, hiding in reservoirs such as the brain, lymph nodes, and gut. These reservoirs are areas where the virus can lie dormant, evading the effects of the drugs. The virus can lie dormant within certain cells in the body, hidden from the effects of the drugs. This dormancy allows the virus to remain in the body indefinitely, ready to reactivate if ART is discontinued. If a person stops taking ART, the virus can reactivate and quickly rebound, leading to a resurgence of the infection. This rebound can cause a rapid increase in viral load, which can damage the immune system and increase the risk of transmitting the virus to others. Moreover, RT requires lifelong adherence to a strict medication regimen, which can be challenging for some individuals and may be associated with side effects. These side effects can range from mild, such as nausea and fatigue, to more severe, such as liver damage and cardiovascular issues. The quest for a cure, a treatment that could eliminate HIV from the body entirely, has therefore remained a top priority for researchers worldwide. Scientists are exploring various strategies, including gene editing, therapeutic vaccines, and immune-based therapies, to achieve this goal. The hope is that one day, we will have a cure that can eradicate HIV, freeing millions of people from the burden of lifelong treatment and the fear of disease progression. Gene editing technologies have emerged as a powerful tool in the fight against a variety of diseases, and HIV is no exception. These groundbreaking techniques, such as CRISPR-Cas9, have revolutionized the way scientists approach genetic disorders and viral infections. By allowing precise modifications at the DNA level, gene editing opens up new avenues for potentially curing diseases that were once thought to be incurable. These technologies allow scientists to precisely target and modify specific genes within cells, offering the potential to either disable the virus's ability to replicate or to enhance the ability of the immune system to recognize and destroy infected cells. This precision is crucial because it minimizes the risk of unintended consequences, ensuring that only the harmful genes are altered while leaving the rest of the genome intact. One promising approach involves using gene editing to remove the integrated HIV DNA from infected cells. This method targets the viral DNA that has been incorporated into the host cell's genome, effectively excising it and rendering the cell free of the virus. This could be a game changer in the quest to find a permanent cure for HIV. By eliminating the viral DNA from these cellular reservoirs, scientists hope to prevent the virus from being able to reactivate and cause a resurgence of the infection. These reservoirs are a significant challenge in HIV treatment because they can harbor the virus for years, even decades, making it difficult to achieve a complete cure. Gene editing offers a potential solution to this problem by targeting and removing these hidden viral genomes. Another approach focuses on editing the genes of immune cells to make them resistant to HIV infection. This strategy aims to fortify the body's natural defenses by equipping immune cells with the ability to resist the virus, thereby preventing it from spreading and causing further damage. This could involve modifying the CCR5 receptor, a protein on the surface of CD4+ T cells that HIV uses to gain entry into the cell. By altering this receptor, scientists can block the virus's primary entry point, effectively stopping it in its tracks. This approach has already shown promise in early studies, with some patients experiencing significant reductions in viral load. Gene editing technologies are still in their early stages of development, and significant research is needed to ensure their safety and efficacy in humans. While the potential is enormous, it is crucial to proceed with caution. Rigorous testing and clinical trials are essential to determine the long-term effects and possible risks associated with these treatments. The journey from the lab to the clinic is a long one, but the progress made so far offers a glimmer of hope on the horizon for those affected by HIV and other genetic diseases. Among the various gene editing technologies, CRISPR-Cas9 has garnered significant attention for its potential to revolutionize medicine. CRISPR-Cas9 acts like a pair of molecular scissors, allowing scientists to cut DNA at specific locations and introduce changes to the genetic code. This revolutionary technology has opened up new avenues for developing HIV therapies that were previously unimaginable. Researchers are exploring the use of CRISPR-Cas9 to target and destroy the integrated HIV DNA within infected cells. This approach aims to eliminate the viral reservoirs that allow HIV to persist in the body despite RT. Additionally, CRISPR-Cas9 could be used to engineer immune cells that are resistant to HIV infection, providing individuals with a long-lasting shield against the virus. While CRISPR-Cas9 holds immense promise, ethical considerations surrounding its use, particularly in germline editing, which could pass on genetic changes to future generations, must be carefully